For attending our soil health session this morning. We will be talking about soil health tests. That we can hand it over to our speaker for the day, who is Steve Coleman. Steve is an associate professor and state specialist in soil fertility at the School of Environment and Natural Resources at The Ohio State University. So I'm really happy to have Steve here and to be with you all this morning. So thanks for being here, and thanks, Steve, for being our guest speaker.
Thanks, Mary. Appreciate the introduction. Let me go ahead and share my screen. Hopefully everybody can see that just fine. Okay, great. Well, thanks again for having me. Thanks for organizing, Mary, and thank you all for being here and spending a little bit of time with us this morning. A lot to talk about soil health. It's great to see so much interest in it, and it's an area that we've been working on for quite a while, but just the interest continues to grow, and so we wanted just to spend some time this morning sharing with you some of the insights that we have. Okay, so I will say that so when we think about soil health, what we're really, the way I kind of conceptualize or to think about it is we're trying to complement pre-existing infrastructure. We already have a lot of information about the chemical processes of soil and really nice commercial soil testing labs and infrastructure that we can go out, we can pull soils, submit them to a lab and get results. And then shameless plug for the new Tri-State, there's documents out there, scientifically vetted experiments and all kinds of research that show what a Mehlich-3 phosphorus value means in relation to fertility. Well, what we're trying to do essentially is to complement that in the context of biological and physical properties of soil. We understand that soil is a matrix made up of these pieces, and all three of these pieces are important and need to be considered, okay? So importantly, when we talk about soil health, we need to think about function, and functions, whatever a process or a piece of that, whether it's nutrient release or availability or drainage or moisture retention or whatever it might be, there's an indicator that reflects that soil function. And I've, a lot of people argue, me included, that if we're really serious about managing soil as a resource and managing some of these functions, then we need to be able to measure it. And this is really a main justification or a call to soil health indicators, and soil health assessment is kind of a standalone science. Lots of indicators out there. We can classify them in, when I say indicator, of course, it's just a measurement or a test. We can classify them as chemical, as physical, as biological. This is a table of, a smattering of things. There's textbooks in soil science have that, they've been writing about methods for decades and decades. This is not any way exhaustive, but there's just a lot of things out there, a lot of ways that we can measure particular processes or components of soil, okay? But when we think about it from a soil health context and from taking it from a research laboratory to your farm, we've got a set of criteria that, some of these indicators or tests that they really need to be. So some of the key things is that they need to be sensitive to management and changes. So if you're going to, be implementing cover crop or doing a no-till or some sort of change in your management, these tests should be able to pick that up. They need to reflect some soil or plant function and that be important in your operation. They should be reliable, robust, and repeatable. So the method should be a repeatable method, something that you're going to get a very similar value if you've run it time and time again. Okay, so that's great. And then we can sort a list of indicators based on that. But then, the reality of a commercial lab comes into play here as well. And it's even a more narrow funnel, and it has to be a rapid test. It has to be relatively inexpensive, and soil labs have to be able to run a lot of them because they're dealing with oftentimes thousands of samples a day. And so, this really kind of filters out a lot of the chaff, and thinking about what are the tests that are available and are being offered or will be offered soon, right? And so no one wants to spend $100 or $150 to run a soil sample to get a soil test analysis. And so we've been very focused here in terms of just focusing on tests that are promising but rapid and inexpensive and capable of running many samples at a time. Okay. And I will stop here, and I just kind of like this slide because, we just need to understand that soil health, especially, management, we understand a lot of things about management, but from a quantification perspective and rolling this out, into a commercial testing framework, there's a lot of refinement that needs, and we're in the very early stages. So I just want to temper everybody's expectations in terms of where we are. Right. One of the things I like to, or a quote I heard that I like to, use is that, why do you think there's a Mehlich 3 soil test extraction? Well, it's because Mehlich 1 and Mehlich 2 weren't good enough, right? And so again, thinking about a successive and refinement in this process, okay? So let's talk about organic matter. It's kind of a big deal, right? If you don't agree that organic matter is a big deal, your Zoom call will be terminated in approximately 5, 4, 3. Now, obviously, we understand, as growers or as scientists or as educators, whatever, the importance and the critical functions that organic matter have and what they actually provide in soil. They really are, it's that small fraction of the soil, but it binds that mineral matrix together, and really what breathes the life into soil as we know it. And as a highly productive soil is a soil with a lot of highly functioning organic matter. And we've known this for a long time. These are two nice quotes that, you can see the dates on them. They're, from a long, long time ago. The decay of organic matter is not, it's not its mere presence is what gives life to the soil. And William Albrecht said in 1938 here, attempting to hoard as much organic matter as possible in the soil, like a miser hoarding gold, is not the correct answer. Organic matter functions mainly as it's decayed and destroyed. Its value lies in its dynamic nature. Folks, who needs coffee when you've got stuff like that to chew on this morning, right? I mean, this is the idea that organic matter, that cycling and that turning over is a critical piece and really what makes a soil healthy, and that's kind of the rub, right? And so how do we measure organic matter? We can do it, and it's done very commonly in soil testing labs. You submit soil samples, you get a percent organic matter. Typically, it's loss on ignition as a method that they run, but there's all kinds of ways we can examine organic matter, not just the total piece of it, but some fraction. So that's really what the rest of the talk is going to focus on, thinking about not the total pool of organic matter, but the fraction, okay? So we can visualize this a little bit as our total organic matter being the full pie here. And that's really what's commonly measured, almost standard in a grower soil test, right? It's like, here's your total organic matter. But we recognize that it takes a long time to build this total pool of organic matter, and it takes a long time to degrade this pool of organic matter. So for a soil health perspective, we're interested in something a little bit more sensitive. Something, organic matter, losses and gains can happen on like the order of decades, right? It might take 10 years to really build it to increase your organic matter 0.5% or maybe a full percent if you're very aggressive about it. But this active fraction, which is a small but very, very important piece of the total, is what we're focusing on as targeting for indicators. And so this is important because this active fraction is really what's biologically associated. It's what a lot of that microbiology is chewing on and turning over and processing rapidly. It's important because where that biology is, that soil biology we fully understand now, and it's really only been in the last 10 years that we understand that role that food web, the soil microbes play in actually processing and stabilizing and building organic matter. So, if we want to think about what's happening or what's the trajectory of organic matter in our soil, this active pool is a really nice place to start, okay? I'll talk about 3 measurements. There's lots to talk about. Really, I'm just going to spend a little bit of time talking about 3 measurements that we focus on in my lab and have been for several years now. Okay, these are what I would call active fractions of organic matter. They're process-based measurements. They involve the biology and the chemistry. We'll talk a little bit about soil physical structure, but really for this talk today, in the interest of time, what we can accomplish in 30 short minutes, we're just going to spend a little bit on this, okay? So respiration. This is a method that most people, it's kind of everyone's often their first introduction into soil health testing. It's a very simple test where we take, a small amount of soil, we add water, we seal it in a jar, we incubate for 24 hours, and then we measure how much carbon dioxide or CO2 comes off that soil. This is a measurement of how much literally soil is breathing, right? What's the respiration rate? So you might have heard of Solvita. Solvita is the commercial example of this, but there's others that are out there as well. This can be run in the field or it can be run in the lab. And there's a few different variations to this. So this is really kind of an introduction a lot of people see, and it's a very important measurement because it's just such an intuitive thing. It's the life of the soil. It's how much that soil is actually breathing and the microbes are respiring, okay? So that's respiration. We'll move on to the second test, which is affectionately known as POX. It's not some affliction that's following on the heels of coronavirus. It's a nice, simple test for permanganate oxidizable carbon. Sometimes this is also called active carbon. This is another simple test. You take soil and a little bit of this reactant solution, you shake it for a couple minutes, you let it settle for 10 minutes, and then you dilute it, and you just read how much this bright purple pigment of permanganate gets reduced or gets consumed by that organic matter. So the more the more transparent, the less purple that's in that sample at the end of that reaction, we actually have more organic matter, okay? So POX is a process pool. It's a very small fraction, 1 to say 4% of the total carbon in the soil. We know it's very sensitive to management. It's a very nice, simple, easy test, okay? And we've done work looking at the relationship between POX or active carbon and respiration. There's no need to measure 2 indicators, if they're actually pointing to the same thing, reflecting the same soil function. But we have a lot of confidence at this point to know that active carbon or POX and respiration actually reflect very different processes. And so although they're related, they're both measurements of this active carbon pool. Active carbon or POX reflects management practices that really promote organic matter stabilization. That's building organic matter, that's building your wood pile, you're stacking wood. And respiration kind of intuitively is breaking down of that carbon and releasing, essentially decomposition, releasing that carbon. When we break down organic matter, of course, we're releasing nutrients with that. And that's what feeds the plant, feeds the microbes, okay? So that's the good stuff in terms of that dynamic nature of organic matter. And we've looked at several long-term trials, both in Ohio and elsewhere, to demonstrate that these pools and long-term management actually do influence how the relative amount of active carbon versus respiration. And I can go into that more, but I won't. I'll just leave it at that for the interest of this talk, okay? The final thing is soil protein. And soil protein is a measurement of, this is a little bit more complicated of a measurement, but there's labs that are getting this online. This is, a method that has been kind of endorsed at the national level. We understand that soil protein is abundant in both plant and microbial biomass. Obviously, we think of protein, we think of, from a human nutrition perspective of nitrogen. It's an important source of nitrogen. Lots of, it's been around for a long time. Proteins have been around. A lot of microbes will degrade it. And it's really like that protein made up of amino acids. The supply of amino acids in soil or depolymerization step is really often the rate limiting step in soil nitrogen cycling. So we believe conceptually that soil protein is a very important indicator of how much nitrogen releases in the soil. And it helps it's like the gatekeeper of how much degradation is going to happen, how much decomposition is going to happen versus stabilization. So let me just skip up and I'll just point to this. We believe these three pieces, respiration as a measurement of microbial activity, POX as a carbon pool, an available carbon pool, and soil protein as a nitrogen pool, kind of work together and help dictate how much that organic matter is cycling, how large that pool is, how fast it cycles. There's a lot of insight that can be gained based on these three measurements, okay? So there's lots of measurements out there. I'm, again, wrapping up here right now, but there's just, a few. We haven't even talked about physical measurements. We can think about things like penetration resistance, infiltration, aggregate stability, bulk density. These are things that can be measured commonly in the field, sometimes in the lab as well, okay? Again, I'm gonna just kind of sidestep this, but if there's questions about physical measurements or any measurement, I haven't talked about the Haney test. There's a lot of things here that we could discuss, okay? I'll wrap up by talking about something I think is very, very exciting, and we're on the cusp here, done a lot of work with infrared spectroscopy and being able to measure the processes that we just talked about, but doing this rapidly and more inexpensively. So the whole game with this technology, it's just, it's infrared, and it takes a reflectance fingerprint of the soil. And based on that, you can see here, there's, different compounds that are there and present. You put it in this magical statistical formula, and it's able to predict properties like soil texture, soil carbon or total organic matter, things like active carbon, protein. We can start predicting properties cheaper. And when we do things cheaper, this might not be the most exciting topic, but when we can do things cheaper, it makes it accessible and makes more people buying into soil health testing. We can roll this out to commercial labs and have this be part of the framework, not just measuring pH and extractable potassium and phosphorus, but actually thinking about some of these other organic matter pools and what the implications are for your management with those pools, okay? Where can I find commercially available soil health testing? We'll talk a little bit more about this as this webinar series moves on throughout the next several weeks. Here's some examples. Brookside Labs here in Ohio have been doing a lot of soil testing. They're not the only ones, certainly. There's university labs. There's things that can be done in field. Again, glossing over this in the interest of time, okay? And then I'll just point to a few resources I think are very good. I'm biased here. We've been working on the soil health website through Ohio State. And of course, Cornell has been a leader in soil health testing, and they do have a really nice manual. I mean, it's a little bit long and there's a lot to dig into, but for those that are very interested in kind of digging into this more, it's a great free resource. So I'm going to end there, and I'm going to, Vinayak Shidakar has some slides as well, but I thought maybe we'll take a few. Okay, Vinayak, take it away.
Right. Well, thank you, Steve. So just to continue on the discussion, as Steve mentioned, the different soil physical, chemical, and biological indicators. One of those indicators, especially the physical and chemical part of the soil properties, is something that we have been dealing with and we're more familiar with. And when it comes to interpretation of soil tests or the soil health tests, and using those for decision making, the biological part is where most farmers would struggle with. As Steve mentioned, there is many indicators that you can choose from, and this is PLFA is one of those many options that I wanted to talk about, because we have some data from the farmers' fields that could be used for this presentation, and it just shows you, gives you a little a better understanding of how these indicators work. So what PLFA really gives you is it's the long form for PLFA is a phospholipid fatty acid. So essentially that's the the compound that the test is going to assess from a soil sample. And based on that, they are able to assess what percent of the total biological, the microbial biomass in that soil relates to fungi versus bacteria, and going further, what type of fungi, and so on. So the basis for this is that a healthier system, which tends to be, we suggest that we don't want it to be disturbed too much. So undisturbed system tends to promote fungal growth more. So that fungi to bacteria ratio tends to be higher in a healthier soil than in a less healthier soil. And it's interesting that how these properties relate to one another. As you have more fungal biomass, the fungi are more efficient in cycling carbon and nitrogen, and that's why if you look at that respiration component, that respiration, the specific respiration from that soil tends to slow down as you have more fungal biomass in the soil. So Steve, if you want to go to the next slide. So just to put all of this in context based on what you would actually see in a field. So this was a small project funded through the North Central SARE grant. And we worked with farmers in Ohio. And luckily we had a site where we were able to sample soils from fields with different history of management. So that number one there is essentially a plowed field with conventionally tilled field with corn soybean rotation, no cover crops. Then we had a field that had recently been put into cover crops, which is that second treatment there. And then the third one was in hay for about 3 years, for about 10 years, which is relatively less disturbance over a long period of time. And then the number 4 and 5, there is not much difference between those, just a few years apart in terms of no-till and cover crops, but they are the longest term fields under no-till and cover crops. So what we did was we sampled these fields and analyzed them for all kinds of soil health properties. And we don't have enough time right now, so I'm just going to present how the PLFA score resulted in. So this is the analysis we got from Ward Lab, which offers a PLFA test with interpretations for about $60 to $80, depending upon who is sending the sample. And the discounted rates and so on. So as you see on the x-axis there, from left to right, you go into a bad management or a not so great soil to a very healthy soil in our perspective in terms of management. And on the y-axis there, you have multiple things going on. So let's look at it one at a time. So if you look at just the bars, the bar graphs where you have that light green and the sort of a fluorescent green portion of that. So as you can see, that fluorescent green, don't pay attention to the numbers yet, but the fluorescent green is the fungi. And as you go from that plowed soil to soil under long-term management of no-till and cover crops, you see that fungal biomass in terms of percentage, which is on the left axis there, goes from 10 to close to 20%. So about a twofold increase in the percent of fungal biomass that goes into increases in that system compared to the bacterial biomass. So that is just the percent. But if you look at the numbers, so the 84 and 739 in that first bar compared to the 620 and almost 2700. So that is if you don't worry about the units, you can see that the fungal biomass, the increase in fungal biomass is almost 7 to 8 folds compared to, let's say, the bacterial biomass, which is about 3-fold increase. So it's not that just fungi are increasing in that soil. Your overall biological constituents in that soil are going to be huge as you go from a conventional system to a very mature system, which you see on the far right there. So it's going to be a huge microbial biomass overall. But the most important thing is that as you stop disturbing that system, you're going to see a greater increase in that fungal biomass. And essentially, what this tells us is how efficient our system is going to be in terms of being able to cycle those carbon and nitrogen in the system. So so the dotted line is at a different scale, but it essentially tells you the bacteria and fungi there is not the only component of that total biomass, but that's what the total PLFA will tell you is total biomass that's in that soil. And if you look at the last 3 bars, the bars on the far right, the 3, there's not much difference you can tell among those 3. And that's where it comes down to in terms of soil health testing. So if you want to go to the next slide, Steve, even not just the PLFA, when you combine all the soil different soil properties into a single soil health score, you do get some indication whether our soil is healthy or not. And so many labs, or most of the labs, would give you a single number that tells you what the overall soil health score is. But I'm sure most soil scientists would agree with this and the farmers, that we don't really know what to do with this number, other than saying that, okay, well, there is this system compared to this system that looks more healthy. So maybe when it comes to soil health testing, it's important to keep in mind that your purpose or the function that you're keeping in mind will dictate what you use. And it's really down to the point where you want to dig deeper into that soil health report and not just rely on something like this, overall soil health score for decision making. You want to actually look into something like PLFA, or your infiltration, or the active carbon, which is more responsive to management, to be able to make some decisions out in the field. I think that's the last slide I had.
Yep. Great. Okay. Thanks, Vinayak. Thanks for that, Sharon. So all that was from Dave Brandt's farm. Is that right? Down near.
That is correct. Not all those are farmed by Dave, but the ones that were under the longest term are from Dave Brandt's farm.
Great.
Thanks, Vinayak, and thanks so much, Steve, both of you, for sharing information with us today. We have actually plenty of questions still to answer, and I'm going to have, as I'm showing the CCA codes up for those of you that want the CCA credits, I'm going to have Ann Shannon, our moderator, who's the Lorain County Extension agent, field the questions to Steve and Vinayak. I will say that we had one comment that now that we've had this overview, it'd be nice to have a little bit more in-depth meeting on some of these things, which is exactly what we were talking about before we got on here. So it's nice to know people are interested in the information. So Ann, if you want to ask some of these questions, I'll go ahead and share my CCA.
Sure. So Steve, one of the first questions that we have is to what can a farmer do on their own farm to measure these measures of soil health?
Mm-hmm. So there are, and others can feel free to join in. There are a number of kind of in-field kits that have been developed. We have one at Ohio State that Rafiq Islam developed. There's NRCS has kind of, that was one of, in my slide, one of those links in terms of, so there's a lot of different options and different tests with kind of instructions. If you Googled something like, USDA soil health kit, you would find that, and there's, kind of outlined protocols or instructions that walk you through what to do step by step. A lot of that can be more from it's really important, I think, for growers to kind of be able to see and internalize the changes that are happening in soil, but a lot of from kind of a precision and a management perspective, I'm biased towards sending soil in and having it done in a lab context, because we can get much better, more reliable and repeatable information that way. So there's a hybrid here, and as this kind of whole field develops, we certainly encourage people to kind of go out and play in the soil and do some of their own kind of testing and just seeing the values that you get and monitor that. But I believe at the end of the day that the true value will come when we develop the infrastructure from a laboratory perspective, the same way that it's gone with soil chemistry.
All right. And that really feeds into that next question. So in general, what can a farmer do to mark their soil health journey so that they can actually document it?
Yeah, that's a great question. I love the phrasing, and that's really how we think about this at this point. Those tests that we talked about, we know that the PLFA, the POX, the respiration, we know that they're sensitive to management, that we know that they're meaningful. Do we know what it means in context of how many pounds of nitrogen are going to get mineralized on your farm in any given year? Like that, we're not even close to being there. And so the thing that we need to do, I believe, is using these tests as kind of a stake in the ground. Here's our baseline, or here's where we're starting from, and using that to monitor what's happening. So it's this idea of it being a journey, this idea of, soil management and trying to tweak things and refine things and understand better what's happening in your soil, how your management is influencing these processes. So I think more concretely, to answer your question, Jason, is get with a county educator, get with us. We're not a commercial testing lab here, but we're running samples through eFields. We'll do a complete soil health analysis, and in partnership with kind of eFields and on-farm research. And then you can see where you're at in 2021, and then you can test in 2024, 2025, and see where you're at that point. So keeping records, all these things that are very important from a farm management perspective, all apply from a soil health context.
Steve, do we know, so the question was posed, are soil health scores consistent between labs? Do we have enough data yet to support that consistency?
Yeah, everybody loves, an overall soil health score. What's my soil health score? Just put it in a number from 1 to 10 or 1 to 100, right? Different labs and different approaches construct that number differently, right? So you might have different actually tests that you use, and you might have different ways to kind of add that up to equal 100. I don't take a lot of weight in a total overall soil health indicator score because of the fact that it's calculated differently. We're still trying to understand what individual methods reflect and how we use them better, how some of them might work together, but not this whole package. And I just think that it's where we want to go, but and I think there's meaning in it, but it's the fact that labs do it all differently. It means that it's kind of it's a little bit chaotic right now. We need more standardization for that really to be meaningful.
Okay.
If I may chime in here, Steve, we did send the same samples to multiple labs as part of that SARE project. And that's what we found, that, yeah, they are not necessarily consistent across the labs, the soil health scores. And that's why I think one of the recommendations is that if you go with one lab, just stick with that lab for the long term. That might provide you better, reliable results that you could use for decision making than trying to shop for different labs or different analysis.
We're going to be running out of time here very, very soon. One other quick question that was asked was, how and when would you recommend a respiration test be done if somebody wanted to use a SALVA kit? So that must be one of the measures that's out there from one of the
I think Steve answered
organizations.
that one a little bit earlier. So
Yeah,
I'll say we do have to wrap up in a little bit, but one of the questions I saw that I think would be good to address is how long does it take for a grower to build organic matter?
Yep.
It's a great question. Yeah. So, you know, the answer is it depends, as always, right? But the tests that we talked about today, all of them, physical structure often takes a little bit longer, but some of these more biological measurements, respiration, PLFA, POX, soil protein, we've seen differences in 2, 3, 4 years. So the 3 to 4 year mark, if you're kind of making a change, is a good kind of ballpark to when you can expect to see changes, right? It's changes are already happening when you do something, when you change your management or implement a new practice, but it takes a little bit of time for that to process through the system and our methods to be able to detect those differences. So 3 or 4 years, and if we keep that on what would be considered a typical soil sampling timeline, you know, you should be able to see some results at that point.
Thanks, Steve. I want to thank Steve and Vinayak one more time for sharing with us today. And I really want to thank all of you for coming and for asking such good questions. I know there are still some questions that have been unanswered, so I'm going to save the questions in the Q&A and in the chat and send them off to Steve and Vinayak and see if we can get some answers typed up that I can include in the follow-up email to all of you, which I will send sometime within the next week. So again, next week our talk is on, we have some data on whether improving soil health can improve yield. So we're going to hear from Jordan Wade next week. We have a whole bunch more sessions of these, and they're all kind of quick. We're hoping to have some breakout conversations in some of our later sessions where we'll be able to interact a little bit more with you all. So that's our plan. We're kind of learning as we go how to do virtual programs and what people are interested in with these topics. So we're really interested in your feedback. I do have a pop-up survey that will come up when you exit this meeting where you can give us a little bit more feedback about what you want to see in the next couple weeks. And, you know, once this series is over, also what we can do to try to facilitate more conversations beyond this. So here's a couple programs that we have coming up for our agronomy team in the next week, aside from soil health stuff, if you're interested. And with that, I'm going to just thank you all for attending. Thanks, Steve, once again, and ask you if you could please fill out your survey when we leave. That would really help us.